Another type of camouflage that we have in the epipelagic is just transparency. So if there's nothing to blend in with, what if you just make your body clear instead? That's also a way of blending with the background by just being transparent. So we see this in a lot of zooplankton. A lot of zooplankton have very transparent bodies. Also, there are some squids that have very transparent bodies. A lot of jellies have very transparent bodies. So it's something we see pretty commonly in the epipelagic are these transparent bodies so that you're able to just not be seen as you're floating around through the water. So next up, we're going to talk a bit about the food webs of the epipelagic. They are often very complex. Many species eat many different food items. Basically, what's usually the limiting factor here is what you can fit in your mouth. Whatever you can fit in your mouth, you're going to eat. Also, many species, again, start out as like zooplankton larvae and then get bigger over time. So you also have one species that's eating very different foods over the course of its life. So it'll be eating very different foods when it's like a little larval fish than when it's a full grown like tuna. So it makes for these very complex food webs with different stages of different fish eating different stuff, and a lot of arrows going every which way. You can see that here in the complexity of this food web. Now, epipelagic food webs often are more efficient in their energy transfer than some other food webs. So I talked about how usually it's around 10% of the energy gets passed from one level to the next. Here it can be a little bit higher. And that also allows for longer food chains to form. So usually food chains are limited to like 4 or 5 steps. And here you can have more like 6 or 7 steps from primary producers all the way up to the final consumer. So you can also tend to get some longer food chains in the epipelagic because of that increased energy transfer efficiency. A member of these food chains I want to mention are forage fish. Forage fish refers to small fish species, so think like maybe like this big, maybe up to like this big. So relatively small fish that primarily feed on zooplankton. So sardines and anchovies and mackerel are all classic examples of forage fish. They're often like kind of small, kind of silvery. They often form schools. And they are often part of what are called wasp waste food webs, where basically you've got phytoplankton, right? Then you've got zooplankton. But most of the zooplankton are pretty small. And then you've got these larger species, things like birds, tuna, snappers, dolphins, etc. You've got these larger species. And often in the middle, between those two levels, you'll have only a few species of forage fish. So you might have like sardines, mackerel, and like squid, and that's it. So you have like this diversity of larger predators, this huge diversity of zooplankton, but there's just a few species that are like the link between those two. And often they'll have top-down effects on zooplankton and bottom-up effects on all those predators. So the effects of the food chain kind of spread out from these few species. So it can be often very important to pay attention to those few species because if something happens to them, the whole food chain can kind of start collapsing. So very important to pay attention to forage fish. Let's look at the productivity in the epipelagic. Again, as I mentioned earlier, most of the epipelagic is very low nutrient water. So this is a map showing nitrate, which, as I mentioned a few lectures ago, is usually the limiting nutrient in the ocean. It's usually nitrate. And you can see how a huge part of the open ocean is very, very low nitrate. So you have low nutrients, nitrate's usually limiting. So because of this low nutrient water, you usually won't have things like diatoms here. You'll probably have cyanobacteria, maybe some dinoflagellates. So these smaller plankton that can survive on lower nutrients. And the microbial loop is very important because the recycling of those nutrients makes a big difference in these low nutrient environments. Another thing to look at is the three-dimensional structure of the area we're talking about. So, we're not just talking about the surface, we're also talking about, the z-axis too. So we're talking about depth. In general, nutrients increase with depth. So you can see this is the Atlantic, Pacific, Indian Ocean on these three different lines. On the left here, this is nitrate. So you can see nitrate is very low at the surface and it kind of increases as you go deeper, and then it kind of stays the same once you're in very deep water. And this is phosphate. Same thing for phosphate. Very, very low at the surface and then increases with depth, and then it's kind of at a solid level as you go down. So why does this happen? It's because at the surface, these nutrients are being consumed by phytoplankton. So if nitrate's limiting, any nitrate that's there, the phytoplankton go suck it right up so they can make more phytoplankton. So yeah, nutrients at the surface tend to be very low because phytoplankton are constantly consuming them. But as you go down, eventually you get out of the photic zone. So now there's no longer enough light for photosynthesis. You have less and less and less phytoplankton. But you also have these little sinking organic particles, things like fecal pellets, larvation houses, dead stuff. All that stuff sinking from the surface. And as it's sinking, you have little bacteria, fungus, etc., colonizing these little particles and decomposing them back into nutrients. And so that is why these nutrients are increasing as you go down, because you have less phytoplankton and also this increased decomposition, putting nutrients back into the water. So that is why it tends to increase with depth. These nutrients tend to be higher in deeper water and very low at the surface. So, kind of mentioned this, but chlorophyll is a proxy for phytoplankton biomass. So a lot of the time we don't measure phytoplankton directly because of course you can't see them with your naked eye. And it's kind of annoying to like grab a bunch of phytoplankton samples, like put them under a microscope and like count diatoms. That's going to take a long time. You're not going to be able to get like a wide area of data from that. So a lot of time what we do is we measure chlorophyll because we can measure chlorophyll with fluorescence, basically with light. So we can use things like a fluorometer on a CTD, or even like satellite. We can get chlorophyll data from satellites as well. And since the vast majority of the chlorophyll in the ocean is in phytoplankton's bodies, by measuring chlorophyll, you can indirectly measure how much phytoplankton is out there. So a lot of time we measure phytoplankton by measuring chlorophyll. So if we were to measure chlorophyll in a vertical profile, so say we have our little CTD and we send it down and we're measuring chlorophyll the whole way, we bring it back up, measuring chlorophyll the whole way, what would you expect that vertical profile to look like? Where would you expect chlorophyll to be high and where would you expect it to be low?